All right. Oh, just met for the pre-lab lecture today, Wednesday, September 25th. And I tried to record the pre-lab lecture and it did not work. So here I am going to do the lecture one more time from my own computer, and I'm going to record it once again, and I'm going to share it with those of you who were not present at the lecture today. Um, So for those of you who are not present, to get credit for today's lab, you have to watch this video and answer the questions and get them correct, and that will give you half credit. To get the other half credit for today's lab, you will have to do the virtual photosynthesis and respiration experiment that I will be sharing at the same time that I am sharing this video. That out of the way, let's jump into the pre-lab lecture. Let me share my screen. Okay, screen is shared. The first thing I want to do is show you where you can find the PowerPoint for this pre-lab lecture. And if you scroll down here, or even, I guess you could skip over here, whichever way you want to do it, and go to Lab 5, Photosynthesis Respiration. And as you can see, again, this is a 2-week lab. So today, we didn't do any experiments, right? Today was only the pre-lab lecture. And that's what I'm about to give you right now is the pre-lab lecture. The first word for attendance in this pre-lab lecture is going to be report. So the first word for attendance is report. At this point, I'm not sure if I'm just going to do this like I do most of the lecture videos, which is a situation in which you just send me the attendance words, or if I'm going to make a Google document or a Google form where you fill out questions while you watch the video. So we'll see. But anyway, the first word for attendance is report. So back to what we're talking about. If you go here, WVSU Online, Lab 5, you can see here the PowerPoint is right here. So let's take a look at it. Let's see. And you know what? I've only used, there we go, slideshow. Will this work? Yeah, there we go. All right. So here we go. Let's jump into this. As I already mentioned, today's lab grade for this week is based on attendance. And if you're watching this because you have to, that means currently you have a 0 out of 20 because you were not in lab. So you need to, at the very least, send in these attendance words. And again, like I said, possibly there might be some questions to go along with it to get half credit, right? Now this is for the people, whoops. What you see here, that's for the people who made it. For you, again, you're going to get half credit, up to half credit for this video. And then you'll get the other 10 points for doing the virtual experiment. So let me know if you have questions on that. Next week's lab is going to be just the regular old thing, right? So you're going to have 5 points for the pre-lab, which is due this Friday. And by the way, I want to point this out. Don't forget, this pre-lab has 2 videos to watch, not this video that you're watching right now, but the pre-lab has 2 videos to watch. Make sure you do those before you do the rest of the pre-lab. And then when you're done with the pre-lab, also part of the pre-lab includes doing the hypotheses. So make sure you do all these before Friday. As always, let me know if you need help. So yeah, that's the pre-lab. And as usual, next week when we meet for lab on Wednesday, you're going to fill out the lab worksheet, and you know that is worth 15 points as it usually is. And then 2 weeks from now, the lab report, not 2 weeks from now, but 2 weeks from the lab, the date of the lab, your lab report will be due, and that'll be worth 20 points. So in a sense, this one lab is worth 60 points, right? 20 points for today's pre-lab lecture, the regular old 20 points for next week's experiments, and then 20 points for the lab report. So let's keep moving. The next word for attendance is going to be lab. Right there, you can see it. So in this lab, we are answering a number of questions. The first one is, does the rate of photosynthesis increase as light intensity increases, right? So we're asking, does the amount of light affect the rate of photosynthesis is basically what we're asking in one of the experiments. And the other one, do photosynthetic organisms respire when light is absent, right? So this one's not about photosynthesis. This one's about respiration. So the question is, do photosynthetic organisms, specifically algae in this case, and I'll explain that later, do they do respiration when they're in the dark? That's what that's asking. Third question, does an increase in temperature result in an increase in the rate of photosynthesis? So again, or another way of asking that is, if we increase the temperature, will that cause the rate of photosynthesis to increase? Will it cause the rate of photosynthesis to decrease? Or will it have no effect at all, right? That's another question that we're answering. Next question is basically the same thing, except now instead of asking about photosynthesis, we are asking about respiration, right? So does a change in temperature affect the rate of respiration, basically? And then finally, the last question is, which colors of light result in an increase in the rate of photosynthesis? And again, another way you could ask that question is, which color is best for photosynthesis, or which color is worst for photosynthesis, right? So we're going to have different colored lights, blue lights, green lights, white lights, and I'll explain all that later. So these are the questions that we will be answering in these experiments in this lab. So how do we answer these questions? That's what comes next. How do we answer these questions? How do we measure the rate of photosynthesis and respiration? And I'm going to bring this up again later, but think about it. If we were doing an experiment for a whole semester, as opposed to just one lab period, which is about 2 hours, right? Let's say we had 16 weeks to do an experiment. How could we measure the rate of photosynthesis and respiration? Well, photosynthesis will be pretty simple, right? We could grow plants, right? And to measure the rate of photosynthesis, we could just measure how much they grew. Like, how tall did they get? How wide did they get? How much weight did they gain? How much thicker did the main stem get? How many leaves did they produce? Things like that, right? There's all kinds of ways to measure the rate of photosynthesis. And we'll talk about respiration here in a second, but let's give some other examples here. How will we measure the rate of photosynthesis and respiration? Well, before we talk about that, there's some things to consider here. First of all, we need to think about the chemical equations, right? So we know photosynthesis uses water and carbon dioxide to produce glucose and oxygen. Meanwhile, respiration is the opposite, right? Respiration uses glucose and oxygen to produce carbon dioxide and water. So how can we measure the rate of photosynthesis and respiration? Well, regardless of what we're talking about specifically, this is giving us a lot of options, right? If we could somehow measure the amount of water, right? We know if water decreases, then photosynthesis is happening because photosynthesis uses water. If we could somehow measure the amount of carbon dioxide, we know that if carbon dioxide decreases, that photosynthesis is happening because it uses carbon dioxide. If we could somehow measure the levels of glucose, we know if levels of glucose are increasing, the photosynthesis is happening because photosynthesis produces glucose. If we could somehow measure the oxygen levels, if oxygen levels are increasing, we know that photosynthesis is happening because photosynthesis produces oxygen. And then really quickly, because I'm saying basically the same thing, but in reverse, if we could measure the rates of the levels of glucose, we know if the levels of glucose are decreasing, then respiration must be happening because it uses glucose. If we know oxygen is decreasing, we know respiration is happening because it uses oxygen. If carbon dioxide is increasing, then we know respiration is happening because it's producing carbon dioxide. If the levels of water are increasing, then we know respiration is happening because it produces water, right? So with all that being said, let's move forward. And I'll give you another equation, something else that's very handy. And you'll need to know this to understand how the experiment's working, and also because you'll need to write about it in the lab report, which I'll bring up later. Here we have carbon dioxide mixed with water. It gives us something called carbonic acid. As far as I'm concerned, you don't even need to worry about the fact that it's carbonic acid. Here's what you need to know. When you mix carbon dioxide and water, it makes acid. So remember what I was saying earlier. If we could somehow measure the levels of carbon dioxide, then we could measure the rate of photosynthesis and the rate of respiration, right? Because if photosynthesis is occurring, carbon dioxide is being used. So carbon dioxide should go down. If respiration is happening, it's producing carbon dioxide, so carbon dioxide should be increasing. Now, add to that this other information that I just told you. When you mix carbon dioxide and water, you produce acid. So once again, if photosynthesis, we'll start with respiration. If respiration is happening, you are producing carbon dioxide, which is mixing with the water, which is producing acid. Conversely, if photosynthesis is happening, you're taking carbon dioxide out of the solution. Therefore, there's going to be less acid. So with all that being said, again, photosynthesis reduces carbon dioxide because it's using carbon dioxide, which is going to reduce the amount of acid in the solution. And as you should know from lab 2, that increases the pH, right? When you remove acid from a solution, when it's becoming less acidic, the pH is going to go up. Therefore, when photosynthesis is happening, we should see an increase in pH, right? So that's what we know. It's very important. You need to know that to understand these experiments, and you'll need to write about it in the lab report. Conversely, respiration increases carbon dioxide levels, right? Because it produces carbon dioxide. Therefore, that's going to increase the amount of acid. Therefore, that's going to cause the pH to decrease. So this is very important. Obviously, everything I just explained is very important. Excuse me, I just heard a noise. Let me back out of here and make sure I'm still recording. There we go. I'm still recording now. Okay. Let me bring this back. So yes, everything I've told you is important. But if you remember nothing else, remember that if the pH increases, photosynthesis is happening, and if the pH decreases, respiration is happening. Now, I also want to point out there's some things to consider here, right? There's at least 2 things that might cause the pH to stop increasing when photosynthesis is happening, right? So again, when photosynthesis happens, pH is expected to increase. But there's 2 things to consider. First of all, there is a limit to how much the pH can increase, right? It can only get up to 14. As a matter of fact, the thing you're going to use to measure pH doesn't even get that high. So it is quite possible that photosynthesis, your pH will increase and then stop increasing. And maybe the reason that stopped increasing is because you've just reached the max limit that you can record, right? So photosynthesis might still be happening, but because of the pH indicator, the thing that we're using to measure the pH, isn't able to measure that change, then we wouldn't know, right? So again, the pH might increase to a point where it stops. And that's one of the reasons why that might happen, right? Because we just can't measure any higher. Some other things to consider. What else might cause the pH to stop increasing, right? So again, photosynthesis is causing it to increase, and it's going to keep increasing. And then theoretically, at one point, we might expect it to stop increasing. And it might not be because the light is intense or not intense enough, or it might not be because the color of the light is yellow instead of red. You know, it might not be because of the experimental conditions. It might stop increasing because of this right here. Let's look at this, right? Photosynthesis requires water. Now, in this case, it's not going to run out of water, so we don't need to worry about that. But photosynthesis also requires carbon dioxide. So as this thing is photosynthesizing and using carbon dioxide, and it's sealed up in this little tube that I'm going to explain later, it could run out of carbon dioxide. So it might stop photosynthesizing because it ran out of carbon dioxide. So it might not have anything to do with your experimental condition, right? It might not be the temperature, if that's what your experiment is about, or the light intensity, if that's what your experiment is about. It might have maxed out because it simply ran out of carbon dioxide. Likewise, for respiration, we do expect it to decrease, but there might be a point to where it just stops decreasing. First of all, again, like I said previously up here, the thing you're using to measure the pH doesn't go all the way down, right? So there's a point where the pH might still be decreasing, but you have no way of knowing it because you can't measure below a certain point. So that's part 1. Part 2, very similar to photosynthesis. Respiration requires glucose. Respiration requires oxygen. And these things are sealed up. So it might be respiring, and then it might stop respiring because it ran out of glucose. Or it might stop respiring because it ran out of oxygen, right? And again, that's not necessarily because of the experimental condition. It may not have stopped respiring because of the temperature that it's in, or the color of light that it's in, or the light intensity, right? It might just be because it ran out of glucose or oxygen. So keep that in mind. And then we'll move on to the next slide. The next word for attendance will be acid. As you can see right here, acid is the next word for attendance. So like I said, these things are sealed up, right? So let's talk about how we're going to measure the pH. So we have these little cuvettes. Yours are going to look different. They're not actually little, they're about the same size, but they're not round. Not that that matters, but you're going to have little sealed up plastic containers that have algae beads in there and water and a pH indicator. And you guys are already familiar with the pH indicator. It's the same thing we used in the second lab where we were looking at the effect of buffers, right? Remember we had two different beakers, one with a buffer, one without, and we added acid to them, and we added base to them, and we saw how the pH changed, and we compared the color of the liquid to this strip right here to determine what the pH was. And that's what you're going to do here too, right? It's the same thing. We are going to put these different algae in their containers under different conditions, and we're going to check their pH every 5 minutes to see if the pH is increasing or decreasing or not changing. But why, let me ask, well, no one's here to, I'm kind of rhetorical, I'm talking to myself here, but I asked the question to your classmates, why do we take measurements every 5 minutes, right? Why not just take measurements at the beginning and at the end? And let me try to show you. Let me see here. I might not be able to do this, and I don't want to waste a lot of your time. Yeah, I can't do that. Let me try one more thing, because I want to make this abundantly clear. I was able to write it on the whiteboard in class, but because I'm making this video separately, I don't have a whiteboard to write on. So let's see if this will work out. No. Again, I apologize, but not to be rude, but when you miss class, sometimes you just got to suffer through things like this. This used to have an option for writing stuff, but I don't see it. Q&A recording. Man, let me just try one more thing. Bear with me here. Online whiteboard. Maybe that. See if this works. Sponsored. That means I probably have to pay for it. Yep, sign up for free. Once again, I apologize that you're having to deal with this, but had you been there in person, this would have worked. Come on. Oh my goodness. Oh, come on. All right. Well, you know what I'll do? One last thing. I apologize. Let's just look at the actual lab. This might help. 101 labs. Photosynthesis respiration right here. Okay. So again, the question that I'm addressing here is, why do we take measurements every 5 minutes? All right. So let's take a look at this experiment. There we go. Let's say, and we're only going to look at two here. Let's say this one started at, I don't know, a pH of 5, and this one also started at a pH of 5, and this one ended at a pH of 9, and this one ended at a pH of 9. Now let me ask you, which one saw the most photosynthesis, right? Was it the one that started at 5 and ended at 9? Let me zoom in a little bit there. Or was it the one that started at 5 and ended at 9, right? You can't tell. If we take measurements at the beginning and the end, that doesn't give us enough information. But let's say this one went 5, 6, 7, 8, 9, right? And then it just stayed at 9. Let's just say it plateaued at 9. And like I mentioned earlier, that could happen, right? It might plateau, not because it's not photosynthesizing anymore or because there's something wrong with being this close to the light. It might be because it just ran out of carbon dioxide, right? So it might be the fact that, yeah, we'll just leave it at that, right? So it might be plateauing simply because it ran out of carbon dioxide. Now, meanwhile, this one may have gone 5 to 5.5 to 6 to 6.5 to 7, 7.5, 8, 8.5, and then 9, right? So now that we have this data, this in-between data, now if I were to ask you which one saw the greatest rate of photosynthesis, the one 10 centimeters from the light or the one 15 centimeters from the light, well, now you know it was the one 10 centimeters from the light, because yes, they both started and ended and had the same total change of 4, right? But we can see that the one closest to the light had a quicker change. So hope that makes sense. Let me erase everything I just did because this is the master copy. There we go. Now back to the slideshow. So that's how we're going to do it, right? In general, no matter which experiment you're doing, that's how you're going to measure the rate of photosynthesis and respiration. Every 5 minutes, you're going to check the pH and you're going to put it in your table. And that's it, right? And now you know why we're doing it every 5 minutes and not just at the beginning or the end. Next slide. So once again, we have these cuvettes that are filled with a pH indicator that you're already familiar with from Lab 2, and some algae beads. Also, let me point this out too really quickly, because I mentioned this to the students who are in person. You might also ask yourself, why do we have multiple algae beads in each one of these containers, right? In each one of these cuvettes. Why not just have one algae bead? Well, there's a couple reasons. First of all, if you only had one algae bead, like say, for example, in this tube, you only had one algae bead, and you put it under light and it didn't photosynthesize, you might be like, oh, well, there must have been something about that condition. Maybe it wasn't close enough to the light, right? But in all actuality, you might have a dead algae bead, right? So you might think it didn't photosynthesize because the light wasn't enough, or the temperature was too cold, or it was the wrong color light, or whichever experiment you're doing. But in all actuality, it may be because you just had a dead algae bead. That's one reason. The other reason is, obviously, the more algae beads you have, the more carbon dioxide is being produced during respiration, and the more carbon dioxide is being used in photosynthesis. So with having multiple algae beads, we're going to see a greater and quicker change in our pH. So now you know. Anyway, again, we've already talked about this. These are the questions that are going to be answered in this lab. So let's talk about them individually. How are we going to answer these questions in this lab and these different experiments? All right, the first question is basically light intensity, right? How does light intensity affect respiration? Well, we're going to have one cuvette in the dark. That's going to be our control. We should expect it to decrease. If the pH does not decrease, that might indicate that all the algae beads in that cuvette are dead, right? Or there's something wrong with the pH indicator, right? Either way, that tells you that there might be something wrong. And if there's something wrong with that cuvette, then there might be something wrong with all these cuvettes, in which case all your results might be invalid. But we'll cross that bridge when we get there, and I'll also talk about that later in this video. So yes, we will have one in the dark. Then the plan is we're going to have one cuvette, filled with algae beads. It's going to be close to a light. Then we're going to have another one that's going to be even closer. And then we're going to have yet another one that's going to be the closest to the light, right? So we're going to have at least 3. I say at least because if I can get my hands on more, then we'll have even more, right? We'll do even more. And also there's a chance that we might run out of materials, and we might only have 2 of them, like one close to the light, one farther from the light. Who knows? But anyway, this is the plan, right? Basically, we're going to have cuvettes from varying distance to the light, and we're going to measure their change in pH every 5 minutes to see which one increased the most, or using that example I used previously, if it happens to start and end at the same pH, so it increased the same amount, then we're going to look to see which one increased the quickest. So that's that experiment. The next one's also pretty simple. The next question we're asking is, do photosynthetic organisms respire when light is absent? So basically, when you put them in the dark, does respiration happen? That's what we're asking. So very simple. We're going to have one in the dark. We're going to have one in the light. The one in the light, in this case, that one is our control. So again, the one in the light, we should see it increase because photosynthesis should be happening. If we don't see an increase in this cuvette, the one in the light, that means there might be something wrong. Just like I said previously with this experiment, right? The first experiment, the fact that it doesn't increase might mean the algae beads are dead. That might mean there's something wrong with the pH indicator, right? So whatever the case may be, if the control, in this case, the one on the light, does not increase, that indicates there might be something wrong, and therefore the rest of your results might be invalid. But again, we'll cross that bridge when we get there. Anyway, the other cuvette is the one in the dark, and this is really the one we're studying because we want to see if things respire in the dark, right? So again, we'll measure these pHs every 5 minutes. And we want to see, basically, does the one in the dark decrease? It's that simple, right? Does it decrease? Yes or no? And again, the one in the light will be the control. So next experiment. Does an increase in temperature result in an increase in the rate of photosynthesis? In other words, does temperature affect photosynthesis, right? As we increase temperature, will that make photosynthesis increase, decrease, or stay the same, right? Those are all different ways of asking the same question. This one's also very simple. We're going to have two cuvettes, both of which are going to be in the light, because again, this is a photosynthesis experiment. One of which is going to be at room temperature, and one is going to be on ice, right? And we're going to measure the pH every 5 minutes. For both of them, we expect them to increase, and maybe one will increase more than the other, in which case we'll know if temperature has an effect on the rate of photosynthesis, right? Because if they have the same change in pH, then we can say temperature does not have an effect. But if one increased in pH more than the other or quicker than the other, then we can say yes, temperature did affect the rate of respiration. Before I move to the next slide, the next word for attendance will be liquid. Liquid is the next word for attendance. Also, I should point this out, and I really could be saying this for all the experiments, but I'm only going to use this example for this one experiment. But again, what I'm about to say could be applied to all the experiments. We could make this experiment better, right? So again, we have one at room temperature and we have one on ice, right? And ice, as you should know, is about 32 degrees Fahrenheit, or 0 degrees Celsius, right? Room temperature in the lab is usually about 70 degrees Fahrenheit. So think about it. That's only two temperatures. We could make this experiment even better if we got something colder than ice, right? So, for example, freezers are usually at 0 degrees Fahrenheit. I should say usually, but very often they're at 0 degrees Fahrenheit. So that's 32 degrees colder than ice, right? So in theory, this experiment would be even better if we had another cuvette where it was even colder than ice. Or if we had another cuvette, not at room temperature, but slightly warmed, maybe like in a bath of water, and that water is at, 90 degrees Fahrenheit, right? So not room temperature, not too hot, but warmer than room temperature, right? So keep that in mind with all these experiments that you do. Technically, they could all be improved, and that's just one of the ways they could be improved. Moving forward to the next one. So again, this one, we're talking about how temperature affects the rate of photosynthesis. The next experiment is the same thing, except we're looking at respiration, right? So because we're looking at respiration, both of them are going to be in the dark, one of which is going to be at room temperature, one's going to be on ice, and we're going to see, if that has an effect. And just like the other one, just like all the other ones, we're going to measure the pH every 5 minutes. In this case, since we're dealing with respiration, we expect the pH to decrease. So the question is, which one decreases the most, right? The one at room temperature or the one on ice? So that's it for that one. And once again, you could think about the different ways that you could make this experiment better by having maybe more than two temperatures, maybe more than just room temperature and ice. All right, finally, the last one. To me, this one's one of the more interesting ones. Basically, this is asking if the color of light affects the rate of photosynthesis. And after Friday's lecture, hopefully you'll have an answer to that. I mean, you're going to have to anyway. You're going to need to formulate a hypothesis. But after Friday's lecture, hopefully that'll be easier because I'm going to explain how light works with photosynthesis, at which point, again, you should be able to have an answer. But the plan is currently that we'll have 4 cuvettes. One of them is going to be in white light, one in green light, one in red light, one in blue light. Again, every 5 minutes for 45 minutes, we're going to check the pH. Because they're all photosynthesizing, in theory, we should expect them all to increase. So the question is, which one increases the most? I should also point out that, again, this is the plan. We might, if I can get my hand on some extra cuvettes and some extra colors, then perhaps we could do maybe white, green, red, blue, and orange, or yellow, right? Some additional color, right? If I can get my hands on it, then we're going to have other colors. But as of right now, this is the plan. So that's it for the experiments that we're going to be doing. So there's some things to consider, some random notes. And this is a test. This is a question on the pre-lab. So here you go. I'm giving you the answer. The purpose of these experiments is not to see how the pH changes, right? That's not the purpose. Yes, we are measuring the pH. We do want to see how the pH changes, but that's just a tool, right? It's just a tool to measure the rate of photosynthesis and respiration. I want to be very clear on that. This experiment is not about pH. pH is simply a measurement tool, right? Just like if we were to do an experiment for the entire semester and we grew plants and measured how tall they got, right? So the purpose of that experiment wouldn't be height, right? We wouldn't be like, this isn't about centimeters or anything like that. It's about photosynthesis. And we're just measuring the rate of photosynthesis in that case by how much the plant grew, right? So same here. pH is just a tool. And again, like I've already hinted at, how else could we measure pH? And we've already talked about it when I showed you those chemical equations, right? If we could measure the amount of glucose, right? We know if glucose is increasing, photosynthesis must be happening because it produces glucose. If we know glucose is decreasing, we know respiration is happening because it uses glucose. So we've already talked about that. You can reverse the video if you want to look at that again. But yeah, there's all kinds of ways to measure photosynthesis and respiration. It just so happens that this is how we are measuring it. Some other random notes. Again, I've already said this, so I'm reiterating it and you're putting it in writing. No cuvette in the dark should increase in pH, and none of them in the light should decrease in the pH. Like I've already said, now you're just seeing it in writing. If this happens, there is a problem. Maybe there's something wrong with the cuvette. Maybe there's something wrong with the pH indicator liquid. Maybe there's something wrong with the algae beads. Whatever it is, there's something wrong. And I should probably reword this because this is pretty definitive, but it shouldn't be. This reads, therefore, your results are invalid. Probably it should say, therefore, you should consider the possibility that your results are invalid, right? So it is possible. This is something that you would need to consider when you're writing your lab report, and I'll get to that shortly. You would need to bring that up. Like, hey, these are my results, but we did have this one cuvette that did this unexpected thing. So that indicates that maybe there was something wrong with the materials or with the experiment, not the experiment design, but the experiment execution, right? So even if the materials are fine, maybe you did the experiment wrong. So those are just all things to consider. Another random note, while performing these experiments, you need to be thinking, and hopefully you've been doing this all semester. I've been trying to train you to do this, but now I'm officially telling you you should be doing this. Think about how these experiments could be performed in a better manner. So think about the flaws in your experiment, right? You've been answering questions like this throughout the semester, and I put them in. I've put those into those lab worksheets for that reason. I've been trying to get you to think like this. How can this experiment be improved? What are some of the flaws in the experiment? These are the things you need to think about while you're doing these experiments. Because not only will it be a question on the lab worksheet, but it'll also be something that you need to write about when you write your lab report, which again, we'll talk about here shortly. And then finally, this bullet point here, while you're doing these experiments, you should think about other experiments that could be performed to answer the same questions. So again, and I've already addressed this, right? And I used the example of growing a plant for 16 weeks. You could measure how tall it got, right? So instead of just looking at algae beads for 45 minutes, you could look at a set of plants for 16 weeks, so on and so forth. So again, in real science, you test a hypothesis more than once, right? You test it one way, and then if your hypothesis is supported, then you go and you try to figure out another way to test your hypothesis. So again, think about other ways, other experiments you could do to test your hypothesis. Obviously, we're not going to do them but that would be the next step in the actual scientific method, right? Yes, you tested your hypothesis, but if you're doing it correctly, you would find another way to test it and keep going. All right, so that's it for that. Now let's talk about how to write a lab report. First thing I'll tell you, because you might be wondering, why do I have to write a lab report? None of you in this semester anyway are biology majors, except maybe one of you. But let's just, for the sake of simplicity, say none of you are biology majors, right? So that you must be wondering why you have to write a lab report, because you'll probably never have to do it again in your life. Well, the first most basic answer is because it's part of the course. That's almost like when a kid says, "Why do I have to do that?" And the parent says, "Because I told you so." Yeah, that's basically that first bullet point. Except I should point out, it's not me that's saying I told you so. It's my bosses. Right? So people much higher than I am, position that I'm in, have decided that lab reports, or at least one lab report, is part of a proper lab section. So there you go. We have to do it. That's part one. The next one is to truly understand the experiment. So you guys have been doing labs all semester. You've been filling out your lab worksheets. Technically, you kind of fake your way through those lab worksheets. As long as you have your data, you could not truly understand the experiment and kind of answer the questions and do a decent job. But to write a lab report, you actually have to really understand the experiment to write about it. So in a perfect world, I would actually have you guys write lab reports on every experiment instead of doing those lab worksheets. But it's not a perfect world. That's too much to expect from a 100 level non-biology major course. So that's part two. And then finally, in my opinion, this is a very important one. Basically, a lab report is like a light version of a peer-reviewed article, right? So all the things that hopefully one day you will be doing your own research and you'll be reading research articles. Well, to help understand them, it helps to have practice writing one. So again, it's not quite the same. A lab report's definitely not the same as a peer-reviewed article, but it's similar, right? So when you're getting a little bit of experience writing a lab report, hopefully that'll help you actually understand some research articles. And the experiment, the example I gave in the lecture that you missed was a disease, right? Like let's say you or someone you love gets diagnosed with a disease. So now you want to do some research on it and you're reading all these peer-reviewed articles about that disease. Again, hopefully because you had this experience writing a lab report, you'll be a little bit more equipped to understand these peer-reviewed articles that you read. So that out of the way. Now we know why you have to write a lab report. Let's talk about how to actually do it. First of all, do not use the internet, right? And what I mean is when you're sitting down at your computer, like, all right, I'm going to write my lab report right now. Do not Google how to write a lab report. The reason is because there's all kinds of different ways to write a lab report. And you need to write a lab report the way I've told you to write a lab report. And this is similar, let me back up, to research articles. So when scientists do research and they submit their research to different peer-reviewed articles, journals, each journal has a different set of rules, and it's really strict. It's funny how strict they can be. Like they'll tell you exactly what font you need to be in, and how much space needs to be in the margins, all these things, right? And they're all different depending on which journal the scientists are submitting their work to. And the same is for you, right? So there's all kinds of different ways to write lab reports. I'm going to teach you how to write a lab report. You need to do it the way I said it, so do not use the internet. Okay, the other one, only write about the experiment that you did. Oh, I need to update this too. There's not 4 experiments, there's 5, right? So 5 experiments will be done in lab next week. You are only going to do one of them. And whichever one you did is the one you're going to write your lab report about, right? You're not going to write about all of the labs, which is good for you, right? It's a lot less work. So hopefully you didn't know that, and hopefully that is good news to you. Third part, it should be past tense and in first person. Why? Just because I said so, right? So, for example, when you're writing about materials and methods, you'll say, I put a cuvette under 10 centimeters from a light source. I put another cuvette 15 centimeters from the light source. I put another cuvette 20 centimeters from a light source, right? Whatever it is, it's you, it's first person, it's past tense. So you don't want to say, we put a cuvette on the ice, or you don't want to say, the first thing you need to do is put a cuvette under a lamp, right? So it's past tense, first person. The next thing, it needs to be double spaced. You should be already familiar with this from independent work, though a lot of you haven't even started independent work, which puts you at a disadvantage. But yes, it needs to be double spaced. Here's another thing that might be good for you. Hopefully comes as a relief. There is no minimum as far as words or pages is concerned. So it's not like you have to, I'm not saying like, oh, this has to be at least 3 pages long. Not like that, right? The shorter, the better, as a matter of fact. And I'm going to come back to that. I'm going to show you a rubric. And as long as you meet everything in that rubric, then that's good, right? As short as you can make it while hitting everything in that rubric, that's what you want to shoot for, right? So as I was saying in lecture earlier to your classmates, this is scientific writing. It's not meant to be entertaining, right? So you don't need to be you don't need to use a lot of adjectives. And again, using the example I used earlier, you would say something like, I put one cuvette 10 centimeters from the light. I put the other cuvette 15 centimeters from the light, right? Straight to the point. What you don't want to do is say something, narrate it. You don't want to say, first, I picked up, I looked at the cuvettes and I chose the one closest to the left and I picked it up and I gently placed it under the lamp, ensuring that it was straight and that I could see it. And then I chose my next cuvette and I picked it up with my right hand in between my first finger and my middle finger, right? You don't need to do all that, right? It's not a narration, just straight to the point. That's what you want to do. This next bullet point, you want to turn it in the same way you would turn in pre-labs, I should say pre-labs, not labs, and written independent work, right? So again, well, you guys have done pre-labs, so you're already familiar with how to do it, right? So you're going to use Google Docs. You're going to write it in Google Docs, and when you're ready for me to grade it, you're going to hit the share button, just like you would with labs. And of course, for those of you who have been doing independent work, then you're also familiar with that. So same thing, right? Use Google Docs, share it with me once you're ready for me to grade it. Final bullet point here. And actually, this isn't exactly accurate. This is from an old semester. I need to update it. But basically, you have 2 weeks to complete your lab report. And I'll give you the exact due date once I share this video and once I share your lab report document. But yeah, it's basically 2 weeks. But this is an important thing to note right here. You can still write parts of your lab report before you have the data, right? So technically, you can start writing your lab report now. There's only certain parts that you cannot write until you've done the experiment, right? You can write a lot of it. And then once you've done the experiment, then you can finish it. And we'll address that when we get to it. But that's the first part of how to write a lab report. Let's keep moving. Obviously, it should be free of grammatical, spelling, and typing errors. I'm not very picky with this. Basically, if something jumps out at me, then you lose a point, right? Something like not capitalizing the first letter of a sentence, or not capitalizing the word I, or you saying I seen something instead of I saw something, right? Big things are the kind of things that I would deduct points for. That being said, I should also point out that if you're using Google Docs, at least on my Google Docs, and I would assume it's the same for you, if you misspell something, it should show you. Make sure we're still recording. Excuse me. Yep, we're still recording. Anyway, as an example, let's just go back to this lab again. If you misspell something, it should show up. So when you're writing a lab report, if you put, say you're trying to do photosynthesis. See how I misspelled it? And of course it's not working. It's not working when I'm trying to show it. But yeah, generally speaking, when you make a mistake, it should show up. No, and of course it's proving me wrong, but yeah. There are things you can do. I don't know what's going on with my computer right now, but usually when there's a mistake, it shows up. It'll underline it in red, and I can fix it. But anyway, that's just something to consider too when you're writing these. Try to be careful for that. Anyway, again, should be for your grammatical, spelling, and typing errors. It should make sense. So there should be enough information in your lab report where anybody who you get, anybody with regular intelligence should be able to understand everything in your lab report. For example, and I'm going to explain this again later here shortly. When you're writing your introduction of your lab report, you're going to explain how photosynthesis and respiration cause the pH to change, right? You're going to explain that. Now, if you didn't explain that and you gave your lab report to somebody who has no idea what's going on, they weren't in the lab, you haven't explained anything, that would make no sense to them if you didn't explain it, right? They would look at your results and be like, okay, these people did an experiment about photosynthesis and respiration, and the only thing they're talking about is how pH changed. That makes no sense, right? So there should be enough information in your lab report where it makes enough sense to anybody who's reading it, right? So, and we'll come back to that too in various spots when I talk specifically about how to write different portions of your lab report. Obviously, it should be accurate or correct. So you're going to lose points if you say anything that's not correct. And I do see this a lot. People make mistakes. So again, going back to the second bullet point, right? You need to explain things, right? So for example, you need to explain how photosynthesis and respiration changes the pH. So when you're doing that, for example, if you were to say something like, when photosynthesis happens, that causes the pH to decrease, you would lose a point for that, right? Because that is not accurate. That is not a correct statement, right? Because photosynthesis causes the pH to increase. So again, you lose points every time you make a mistake like that. It should be precise. I won't even talk about that. That's not really an issue really in this course. It should be succinct. I've already talked about that. Again, you don't need a narration. The shorter, the better. Just get to the point. You don't need a bunch of colorful adjectives. You don't need to make it entertaining. Just get to the point. It should be clear that you understood the experiment. This also goes hand in hand with this one. It should be accurate or correct. So yes, you will lose a point for every mistake you make. Everything you write that's not correct, you'll lose a point. And if there's a lot of them, if you make a lot of mistakes, then I'm just going to assume you didn't even understand the experiment. So then you'll lose even more points, right? If you're just writing a bunch of nonsense, then to me, it would seem like you didn't understand the experiment, at which point you'll lose more points. And that's it for that. So I'm going to give you a grading rubric, right? It's not, this also needs to be updated. It's not going to be shared in Google Classroom. I'm going to share it via Google Docs, via WVSU Online. And instead of showing it here, let me show you where it actually is. And again, I'm going to share it with you, but it is already technically available. And let me show you where. If you go to the announcements and you look at that very first announcement, at the very bottom, there's a list of links. And one of those links is a list of every single due date for the semester. If you go here, I'm going to hit Control F because I'm going to search this document and put lab report. There it is. All right. Lab report. You can see here the lab report is due on Friday, October 18th. Not only can you see the due date, but you can see right here, if you click on it, it brings you to where you're going to submit your lab report. And also click on this. And this is the worksheet that you're going to use to write your lab report. So again, do not use the internet to see how to write a lab report. You're going to use this and just go line for line. If you do everything that's in here, you're going to be fine. As usual, just like any pre-lab, you're going to make yourself a copy of this, right? File, make a copy. You're going to share it with me when you're done. But this will be the very top of your lab report, right? And then down here below this, right? Start writing your lab report right here. And again, all you have to do is go step by step and you should be okay. Is there a title? It's like, okay, I better put a title. Is the title appropriate? Okay, I better make sure the title is appropriate. Is the abstract section labeled? Okay, I better label the abstract section. Is it a brief summary of what the rest of the write-up is about, right? So as long as you go through this line by line, you should be okay, right? So it tells you exactly how to write your lab report, and it even gives you the points value. Like if you mess something up right here, for example, does it include an explanation of how the student formulated their hypothesis, right? If you leave that out, you're going to lose a point. It's all very straightforward. Excuse me. There we go. So again, the grading rubric will be shared, and I just showed it to you. You know how to find it, and I'm going to share it again probably when I share this video. And this, as you can see here, is just like a little snippet of what that entire document looks like that I just showed you. And again, that document I just showed you shows you all these things, but let's go through them very quickly, right? These are the different sections in your lab report. Let's talk about them. Your next word for attendance will be witch. Not like this witch, but like a Halloween witch. Like since we have Halloween coming in about a month, right? So the next word for attendance is witch, W-I-T-C-H. Anyway, the first part is the title. Obviously, you need a title. The part that's not obvious is you need to make sure that the title is appropriate for the experiment that you did. So, for example, photosynthesis and respiration is not a good title, right? Because you're not, if you're writing a lab report about all the experiments, that might be a fine lab report title. But you are writing about the experiment that you did. So your title should describe the experiment that you did. Pretty simple. Abstract. This part's a little bit more complicated, especially I think a lot of people have issues understanding the difference between abstracts and introduction. So let's jump into it. The abstract is a brief summary of the entire paper, right? So it's taking the entire paper and condensing it into into just a small summary. As always, it should be labeled. So on your paper, it should say abstract, and then you should write your abstract. It should let the reader know if they want to read the rest of the paper, right? So that's the purpose of abstracts in general. Obviously, when you're turning in the lab report, I don't have a choice. I'm going to have to read your paper whether I want to or not. But in general, that's one of the purposes of an abstract. Like, so when you're doing research and you're looking at peer-reviewed journals, you're going to be looking at the abstract. One, to see if you want to read the entire lab, or sometimes you're just out for information. Like, for example, I might say, I might wonder how the COVID lockdown affected carbon dioxide levels in the world, right? So I would look at that. I would look at peer-reviewed journals, and I would read the abstracts. And if I found a peer-reviewed journal that in the abstracts said the lockdowns from COVID-19 resulted in a 5% reduction in carbon dioxide levels worldwide, then that's it. Then that's the information I needed. I got it right there in the abstract, because I don't need to read necessarily the whole peer-reviewed article to see what it's about, right? I got the results. That's what I was after. So there you go. So that being said, your abstract should provide the same thing. It should include the question being answered in the lab, right? So are you looking at light intensity? Are you looking to see if things respire in the dark? Are you looking how temperature affects photosynthesis or respiration? Or are you looking how the color of light affects photosynthesis, right? You're going to put the question, right? So you'll say something like, in this experiment, I wanted to see if the color of light affected the rate of respiration, right? That includes the question. Then you give a basic description of how you answer the question. So you would say something like, to answer this question, I put algae beads under different colors of light and measured their rate of photosynthesis and respiration, right? That simple. And then also you need the basic results, right? So in that case, you might say, and again, let me just back up and say the whole thing all in one sentence here, or a few sentences. In this experiment, I wanted to see how the color of light affected the rate of photosynthesis. To do this, I put different algae beads under white light, green light, red light, and blue light. And I found that, red light was the best for photosynthesis and green light was the worst for photosynthesis, or whatever it may be, right? So just the basic results, right? You put the question, you basically describe the experiment, not in detail, just the basics, and then the basic results. What color was the best for photosynthesis? What color was the worst for photosynthesis? And again, that's just the one example I'm using. Obviously, there's 4 other examples. So your abstract might sound a little bit different. So that's it for abstract. The next one is introduction. Now, this is the part, before I even pull it up and let's read what the screen has to say. An introduction is the part that provides so much information or enough information to where the whole lab report makes sense. So again, as I mentioned earlier in this video, you're going to talk about the results, right? You're going to talk about how the pH changed. So the reader, before they get to that part where they're reading about how the pH changes, you're going to explain in the introduction what that means, right? So again, the introduction will have enough information to where when they read the rest of this stuff, it will make sense. That out of the way, let's look at what the screen says. Obviously, just like everything else, it has to be labeled. So you're going to have the word introduction. And then under that, you're going to write your actual introduction. Like I've already said previously, it should give enough information about photosynthesis and respiration that the reader will understand the rest of the lab. So at the very least, then, you should discuss how photosynthesis uses carbon dioxide, and you should discuss how respiration produces carbon dioxide, right? Because obviously you're going to need to, and this will explain next, excuse me, I'll come back to that. Because later in the introduction, you're going to need to explain how photosynthesis changes pH and how respiration changes pH. And you can't explain that unless you first explain which one uses and which one produces carbon dioxide, right? So again, you have to have enough information, you give enough information about photosynthesis and respiration to where that will make sense. Then you're going to repeat yourself, and you see there'll be a little bit of repeating yourself in these lab reports. Once again, you're going to include the question that you were answering in the experiment. So even though you already said it in the abstract, you're going to say it again. In this experiment, I wanted to see how light intensity affected the rate of photosynthesis, right? If that was the experiment you did, that's what you would write. Now, for the first time in this lab report, you will also then include your hypothesis and why you chose your hypothesis, right? So again, putting these 2 bullet points together, in this experiment, I wanted to see how light intensity affected the rate of photosynthesis. I hypothesized that the more light there is, the slower hypothesis will be, because I don't know, because plants don't like light, which is not a true statement, but again, you'll have your hypothesis and you explain why you chose your hypothesis. Like, what's your reasoning for saying what your hypothesis is? And I also should point this out too, really quickly. If you look at the lab, it's already there for you. You can copy and paste, right? So here, where's it at? For example, this one. What is your hypothesis? Right. Does temperature affect the rate of photosynthesis? What is your hypothesis for this question? Why do you believe this to be true? So you've already done it, right? So once you've done a lab worksheet, here's your question, here's your hypothesis, and here's your reasoning for the hypothesis, right? So you could just take that, you can literally copy it and paste it into your lab report. Next, again, I've kind of alluded to this earlier, but you're going to explain how photosynthesis and respiration affect pH, right? What is it about photosynthesis that causes the pH to increase? What is it about respiration that causes the pH to decrease? You should explain this, right? So that the reader knows what you mean, what you're talking about when you say the pH increased. And they'll know why you're even measuring pH to begin with. So without explaining this, that would be confusing to a reader, right? So again, you're going to explain how photosynthesis and respiration affect pH. Because again, pH is the tool that we're using to measure the rate of photosynthesis and respiration. There's a lot. Let's talk about the things that it should not include, right? You don't even want to talk about how the experiment is performed. Now, again, in the abstract, you give a basic description, but even then, you don't go into detail. And then here in the introduction, you don't really talk about how you're going to do the experiment. Now, don't get me wrong. Like I just said, you do explain how photosynthesis and respiration changes the pH. So obviously, when you're explaining that, you would mention we're using pH to measure the rate of photosynthesis. This is how photosynthesis changes pH. This is how respiration changes pH. but you're not going to get into the little details. Like I had this many lamps and I placed these things this close to the lamp, so on and so forth, right? You don't want to do that. And you don't want to include the results. So even though in the abstract you did give the basic results, in the introduction you don't give the results, right? You just talk about the question that you're answering. You give enough background information about photosynthesis and respiration. You give your hypothesis and explain why you chose your hypothesis. You explain how pH is going to be used to measure photosynthesis and respiration, and that's it. There's no need for results. So that's it for the introduction. Next is materials and methods. This one's pretty straightforward. This section should give enough information that the reader could perform the experiment from reading it. So when you are writing this, again, just also good time to remind you, it should be first person past tense. The entire thing should be first person past tense. But again, you should write in enough detail when you write this materials and methods section that you could give it to somebody and they could perform the experiment, because you should have enough details explaining it. And when I say enough details, first of all, you should include everything used. So all the materials, right? So you have cuvettes filled with algae beads and water and pH indicator. You have a pH color guide. You have a lamp. You have a ruler, right? Everything that you use, you should mention it. And you should do that while you are mentioning everything that you've done, right? So you need to mention everything. That should be pretty straightforward. I can't say too much about that because, again, I think that's pretty straightforward. What it should not be is copied word for word from the lab manual. First of all, the lab manual is not past tense and it's not first person, right? The lab manual is present tense and is giving instructions like, first, put this cuvette here. Second, put this cuvette there, right? You don't want to do that. Explain what you did, first person past tense. Another thing is to make sure that you write what you actually did, right? So what you did might vary from what the lab manual or what the lab document tells you to do. Again, for example, when you're doing the light intensity one, this one informs you should have 1 10 centimeters close, 1 15 centimeters, 1 25, and 1 in the dark. But this is your experiment. So if you change it, maybe you have 1 at 10 centimeters, 1 at 25 centimeters, and 1 at 50 centimeters, right? I don't know. Do what you will, but just make sure that on your lab report, also, and right here too, make sure that your lab report reflects what you actually did, not what the lab worksheet told you to do. Because again, these are our experiments. We can change them if we want, as long as we account for that in our documentation. All right. And then finally, you should not explain why you were doing what you were doing, right? So when you say that you check the pH every 5 minutes, you don't say, I check the pH every 5 minutes because if the pH goes up, that indicates that photosynthesis is happening, right? You don't do that. You've already explained why you're doing what you're doing in the introduction. So here you just say what you're doing, right? So I recorded the pH every 5 minutes, right? Yeah, so that's it. That's it for materials and methods. Let's look at results. You are already very familiar with results. I've already trained you to do this. Every lab you've done this semester, for the most part, has done this. So if you remember, for every lab you've done this semester, it should look a little bit like this, right? You filled out a table with your data, and then the very next question is basically asking you to repeat the information that's in the table, right? You're going to put the numbers in here into paragraph form. So to give you an example, if you were writing results for this, that one started at a pH of 5, ended at a pH of 9. Let me zoom out. With a total change of 4. And let's say this one started at a pH of 6 and ended at a pH of 9 with a total change of 3. Right, obviously there's going to be middle stuff too, but again, these are your results. You're just going to put it in paragraph form, right? So again, going back here, you're going to list the starting and ending pH of all the cuvettes, right? So the cuvette closest to the light started at this pH, ended at this pH. The cuvette farthest from the light started at this pH, ended at this pH. You're going to include that information. You're also going to include which one increased or decreased the quickest, or the most, or the slowest, or the least. So try to remember the example I gave earlier in this lecture. Sometimes you might have the same total change of pH, which doesn't tell you anything. So in that case, you need to look like, all right, they both increased by 4, but this one increased quicker, right? Or this one decreased slower. So it's not always about just this first bullet point, which is which one, excuse me, it's not always about just which one changed the most. Sometimes it's also which one changed the quickest or the slowest, if applicable, right? So again, one last example. If this was the data, whoops, Control Z. If this was the data, then you wouldn't need to explain which one was the quickest and the slowest, right? Because they all had different. You could just say this one changed the most, followed by this one changed, followed by this one, and this one changed the least, right? So it's not always applicable. You might not always have to describe the quickest or the fastest. Sometimes you can just describe which one happened the most or the least. Let me delete this really quickly because this is the master copy. All right, back to this. It should include which cuvettes didn't change. So again, if you're describing which ones changed the quickest, which ones changed the most, so on and so forth. And obviously, if one of them doesn't change, you're also going to mention that too. When you do this, you need to identify their cuvette by their condition, not their number. So if you read the lab, it instructs you, right? Write number 1 on this cuvette, write number 2 on this cuvette, so on and so forth. But when you're writing about it, you don't say that. You don't say cuvette 1 started at this pH, ended at this pH. Cuvette 2 started at this pH, ended at this pH. You're going to describe the condition. So you'll say something like, the cuvette in the green light started here, ended here. The cuvette in the white light started here, ended here. Right? Because if you just use a number, that's going to make no sense. You need to talk about the condition that the cuvette is in. What color light is it in? What temperature is it in? How much light intensity? So on and so forth. And this is a little bit tricky for some people, but you should not mention photosynthesis or respiration, right? So you're just saying this is how the pH changed. You don't explain what that change in pH means. First of all, you've already alluded to it in the introduction when you described how photosynthesis and respiration affect pH. But again, you're just taking what's in the table, those numbers that are in the table, and putting it into paragraph form, just like you have for nearly every experiment that you've done this semester, right? I wrote those labs for a reason, or like that for a reason. I've been training you to do this, so you already are familiar with this. Speaking of which, that brings us to the discussion. And in a sense, part of a discussion is basically repeating the results, but this time now you explain what it means, right? So this section explains the results and discusses the experiments, right? So first you need to restate the purpose of the experiment. So even though you've already mentioned this, the abstract and some of it in the introduction, you need to say it again, right? In this experiment, this is the question that I was trying to answer, right? You repeat your question. You repeat your hypothesis, excuse me. Yeah, I'll just put these things together. So you're going to, again, repeat the question. You're going to repeat your hypothesis. And even though you just got finished describing the results, now you're going to explain what it means, right? So if this cuvette had the largest increase in pH, now's the time to say, that means this cuvette photosynthesized the most, right? And then once you've explained that, then you can explain how that affects your hypothesis, right? The one that it photosynthesized the most, does that support your hypothesis or does it not support your hypothesis, right? So again, you're re-explaining your results, but this time you're explaining what it means in the context of photosynthesis and respiration, right? And then once you've explained that, you're explaining does it or does it not support your hypothesis. So again, you're going to need to restate your hypothesis, right? I hypothesized that the cuvette in the purple light would photosynthesize the most. And as you can see from the data, the pH increased the quickest and the most. Therefore, it supports my hypothesis, right? It's pretty straightforward. If your hypothesis was wrong, you should explain why you think your hypothesis was wrong. So do you think it was that your hypothesis was wrong? Like, for example, if you think, you know, think of the first experiment. If you think that the cuvette in the dark is going to photosynthesize more than anything else, obviously that's a really horrible hypothesis, because photosynthesis doesn't happen in the dark, right? So in that case, if your hypothesis was wrong, then you would explain why. You'd be like, oh, well, yeah, my hypothesis was wrong because photosynthesis requires light. Or maybe it's not that your hypothesis was wrong. Maybe there was something wrong with your experiment. And that's when you would explain that, right? So going back to the things I mentioned earlier, when we were talking about thinking about different ways you could, the different flaws in your experiment. So keep that in mind, because this is where you might bring it up. So you might say, the data does not support my hypothesis, but it could be because, and then you explain a particular part of the experiment that may have gone wrong that led you to misleading results. And then another big mistake people make is this little bullet point right here. I just want to point this out. This is the case for every single experiment we do this semester, but let's talk about it again. This experiment does not prove that your hypothesis was correct, right? You didn't prove it was correct. You didn't prove it was incorrect. It can only support your hypothesis, right? So when you're writing this, you can say the data supported my hypothesis, or the data did not support my hypothesis. You didn't prove your hypothesis was correct. You didn't prove your hypothesis was incorrect. You either supported it or you didn't. Next, you should address the limitations of the experiment. I've already said that today, and I've already trained you to do that throughout the semester. Every lab you've done this semester asks you this question, right? It says, how can this experiment be improved? What were the flaws of this experiment, right? I keep asking you these things because these are the things you need to be thinking of. So you'll need to address that in this lab report. And also, sorry to do this, but let me back up again. Again, I said here, right, if your hypothesis was incorrect, then you need to explain either why your hypothesis was incorrect, or perhaps maybe the experiment went wrong, right? And that's why your hypothesis, that's why it didn't support your hypothesis, because maybe there was a flaw in the experiment. Again, that's kind of what you're discussing here, right? You're going to talk about the flaws in your experiment. For example, if the results suggest that algae photosynthesize better at more intense light, does this mean that there's no limit to this, right? So that's another thing. When we're addressing the limits to your experiment, the limits to your data, right? So we could find that the cuvette closest to the light did in fact photosynthesize the most. But then think about what that actually means. Does that mean if we were to keep increasing the light intensity, either by bringing the cuvette even closer to the light, or what if we get a stronger light source, right? Does this mean like no matter what, if we keep increasing light intensity, that photosynthesis will keep increasing, or do you think it'll hit a limit, right? So again, you're speculating here, but that's okay. There's things you need to consider. And again, that's just one example, so you can't use that example in your lab report. And then finally, and again, I've been training you to do this too throughout the lab, all the labs you've done this semester, discuss other experiments you could perform to test your hypothesis. So yes, like I said earlier today in this video, in real science, if you want to call it that, you test a hypothesis, and if your results support your hypothesis, then you go back to the drawing board and you figure out a different way to test your hypothesis, right? You keep coming up with different ways to test your hypothesis. And since you're not actually going to do that, what you are going to do is think about other ways that you can test your hypothesis. And for example, rather than observing pH change in a cuvette for 45 minutes, you could put algae in 2 fish tanks with different amounts of light and observe their growth for 45 days. So again, using the first experiment as an example, if you wanted to see how light intensity affects the rate of photosynthesis, you, like it says right here, you could have one aquarium, if you will, fish tank, an aquarium that has a little bit of light, and then you could have another aquarium that has a lot of light, and you could just let it sit there for 45 days. And after 45 days, you could see which one has the most algae in it, right? And that's just one way you could test the same hypothesis, right? And again, that's just an example. Please don't use that example. Just think about different experiments you could do. And in this case, you could even look it up if you want. So that's it for the discussion section. The next one should be pretty quick and easy. It's pretty straightforward. Figures and tables, the next section. Figures, a figure is your graph, and a table is obviously your table. Actually, I probably should have put that in different order just to make it easier for you. But table is pretty straightforward. Actually, both of these are straightforward. You've been doing these all semester. The table, literally, you're doing it right here in the lab, right? So you can literally, if this is the experiment you did, just highlight it. Whoops. Control C, right? Copy it and then paste it into your lab report, right? You can literally just copy it and paste it. It's that simple. The only part that might be a little bit different, excuse me. Let me go back out here. You can see, for example, this is table 2, right? Because this is the second experiment. Now, when you copy and paste it, that part you'll have to change, right? Because this might be table 2 in the lab worksheet, but in your lab report, there's only one table. So therefore, it's going to be table 1. And I hope that makes sense. If not, let me know. Then finally, the next part is the graph. You're already familiar with graphs. You already know how to do this. The only difference between this graph and the graphs you've done previously in the semester is that now you're required to do it electronically, right? Use Google Docs, use Excel, use some sort of computer program. This is the one time you cannot do it by hand. Now, don't get me wrong, you can do it by hand for the lab worksheet if you want, but for the lab report, you need to use some sort of program, and I can help you do that. Yes, so there's that. As always, you need to label it, and again, the first graph will be titled Figure 1, and then obviously you'll give the rest of the title to describe the data in the graph. Now, I will point this out. This is a good time to point this out. Depending on your experiment and your data, you might be able to do multiple graphs, in which case you could get extra credit. So obviously, if you have multiple graphs, the first one will be called Figure 1, the second one will be called Figure 2, so on and so forth. So keep that in mind too. You can get extra credit for doing multiple graphs. And as always, for every graph you make this semester, make sure you name it. Make sure that the name describes the data, right? So for example, a horrible title for a graph would be photosynthesis respiration lab, right? That you would lose points for that. It needs to describe the data. And as always, for every graph, it should include enough information that the reader knows what's on the y-axis and the x-axis, right? There should be enough information. You label your axes, you give it a good title. Anybody reading the graph should understand what the graph means, even if they've never even read the lab report. And as always, part of that would be including units, right? So, for example, if you're doing a line graph, your x-axis would be time. So you would have time, parentheses, minutes, right? Because we measured the time in minutes. Yep, so that's it for figures and tables. This last part is even easier, and we're almost done, and I'm glad because this is, again, the second time I've done this lecture, and I've did them back to back, so my throat's killing me. But literature cited is the last part. And let's say literature is the next word for attendance. Literature cited is the last part. It's very simple. You're just going to list your sources. Now, technically, you don't even need any sources. Technically, all the information in that lab worksheet, or it says lab manual here, but it's a lab worksheet, that gives you enough information to write your lab report, right? But if you happen to use other sources, like in the introduction, when you're giving background information about photosynthesis and respiration, and you want to include more information that was presented in the lab worksheet introduction, which you don't have to, but if you wanted to, just make sure you include your sources in this section. And the only reason I would say this is important is because if there is a piece of information that is in your lab report, like a fact, if you state a fact that's in your lab report that does not come from your lab, then you're going to lose points, right? Because if you have a fact, it needs to be backed up with a source. The format doesn't matter. In a perfect world, you would use APA. That's what we use at this university. But it doesn't matter to me as long as you have a literature cited section and you list your sources. And let me also say this too, and I failed to say this earlier to your classmates, and I guess I should remind them. Even if you don't use any other outside sources, you still need to have a literature cited section, and you still just need to write lab worksheet, right? And that'll be fine. So you still need this section, even if you don't use outside sources. You need to at least list your lab worksheet. And that's it. We are done. The last word for attendance is not going to be a word at all. Instead, well, it will be, but the answer will be different. So the last word for attendance will be your first name. And that's it. If you have questions, let me know. This video, basically, because if you missed the lecture, you can consider this your pre-lab. So this needs to be done by Friday. You need to answer all the questions by Friday. Otherwise, you get 0 credit for today's lab, and you will not be able to come to lab next week. So that's a 0 credit for next week's lab, which means you cannot write a lab report, which means you'll get 0 credit for that. So you potentially could lose 60 points if you do not have this done by Friday. And once again, one last reminder, because you were not in lab today, you have to jump through some extra hoops to get full credit. So this video and the accompanying questions count for 10 points, and the other 10 points are going to come from the virtual photosynthesis experiment that I will share with you when I share this video. And that's it. Let me know if you need help. Sorry this was so long, but it's a very important lab.